Hello, so today we're going to be talking about microscopes. Alright, so some vocabulary that's going to be very helpful for you all while we're talking about microscopes. The first one, obviously, is the definition of microscope, which is an instrument used for viewing objects that are too small for us to see by the naked eye. So it helps us see things that we couldn't usually see with our eyes. Our second vocab word is going to be magnification. Magnification is making an image appear larger than it actually is, so magnifying it to look larger. Resolution is how clear an image is. The field of view is how much area we're able to see when we're looking through the microscope. And the depth of field is the thickness of the specimen that is acceptably sharp at a given focus level. So the depth of field is which level of thickness is actually clear at any given time. So here's a look at magnification versus clarity. So here we go, resolution. They're both magnified the same, but the resolution on image A is much better because the resolution there is much more clear. Image B has a low resolution because it's not very clear at all. But they're both magnified the same. Zoomed in the same amount. One higher resolution because it's clear, and the other one lower resolution because it's not clear. So let's look at this scenario. So Chantel wanted to look at her skin cells under the microscope. After making a wet mount slide, she looks under the microscope and the image she sees is clear, but it's not big enough. Is that a problem with resolution or magnification? Alright, so this one, it's a problem with magnification because it's clear. And again, resolution is how clear an image is. So the resolution is fine, but because it's not big enough, it's a problem with the magnification. So she'll want a higher magnification to make the image appear larger. So there are going to be various types of microscopes that'll help us see some objects either better or actually see something we can't see at all using our eyes. So the dissecting microscope is used during dissections to help see small structures more clearly. A compound or a light microscope is used to see things like cells that we couldn't typically see with our eyes. And then a scanning electron microscope is going to be used to see teeny tiny things smaller than a cell, like viruses, teeny tiny bacteria, proteins. And then a transmission electron microscope is another type of electron microscope that again allows us to see those teeny tiny things. And in a few minutes we're going to talk about the difference between those two. So a dissecting microscope, it uses light to help you see a 3D image. Now they are lower magnification, and the reason behind that is because they're mainly used during dissections to get better views of certain specimens. So it helps you see things a bit more clearly during a dissection, but you're not able to see individual cells. So we don't have an image of that. A dissection microscope typically isn't as large. It's fairly small microscope, and it would be used to see things like the lining of the intestine, to see the structure of it a bit better. Even though I can already see it with my eye, I might not be able to see the fine details. And that leads us to the compound, also known as the light microscope. This is the one typically that you'll see in the biology classroom. And the compound or light microscope uses light to pass through a specimen. So the specimen we look at is very thin. So the dissection scope, we don't necessarily have to have thin specimen, but with a light microscope, we need it to be thin enough for the light to pass through. The light microscope is also known as the compound microscope because it uses a series of two lenses. So you have the lens at the top that you initially look through at the eyepiece, and then you also have a lens down here These are known as the objective lenses. So it's called compound because it uses two lenses to view your specimen through. The microscope is going to make a two-dimensional image, not a three-dimensional image, and their magnification varies from 10 times power to 400 times magnification. So making something look from 10 times larger all the way up to 400 times larger. So that's a typical range of magnification on a compound or a light microscope. And again, these are the main ones that you're going to see be used in the biology classroom. And therefore, they are typically used to view live cells or, in general, cells. So here are our electron microscopes. If you notice, the electron microscopes, they're much, much larger. This is not something that you would see in a typical high school. These are going to be reserved for college settings because they are much more powerful, much more expensive, and much larger microscopes. In the electron microscope, it's called electron microscope because it uses electrons to help form images of objects. And it is 100 times more powerful than a light microscope. It helps you see things like viruses, bacteria, proteins. Sometimes there are even ones that can let you see as small as an atom. The magnification on an electron microscope can go up to 10 million times larger than the object actually is. So it can magnify an image up to 10 million times larger. And these can only be used to view dead cells. So it's going to be, the cells have to be dead in an electron microscope. So here's an example of something you might see using an electron microscope. It can help you see the little nano fibers. So this is the light microscope versus the electron microscope. Again, you can see much, much more finer detail with the electron microscope than the light microscope because it zooms and magnifies much, much larger. There are two kinds of electron microscopes. We have the transmission electron microscope, which uses the beam of electrons to go through a thin section of a specimen. The image is only two-dimensional versus the scanning electron microscope. The specimen is whole, so you get to see the whole specimen. It's often plated in gold, and it bounces electrons off of the sample to create a 3D image. So the main difference between these transmission electron microscopes is only a section of a specimen, and the image is two-dimensional, versus the scanning electron microscope, you get to see your whole specimen in a 3D image. So here would be an example of a transmission electron microscope image, two-dimensional, you can't see terribly many aspects of this, and this is a section of a sperm, versus here we can see the entire sperm in a three-dimensional image. So here we're going to do some what in the worlds. So we have some magnified images here, and let's think about what they are. This one is an eyelash. Can you imagine? That looks so much larger than an eyelash because it's so highly magnified. This one, you can kind of figure out or have an idea of what it is based over here. This is actually paper, highly magnified paper, versus a dust mite in this image. Ooh, these are funny looking. So this is a grain of salt. And over here, this is a human tongue, highly magnified. What do we got going on with these two? A flea. This is the head of a flea. And this is the close-up of its antenna. So it's further magnifying this part here, over here. This is a scanning electron microscope image of a housefly. You can see all the little details. So when we're talking about the light microscope, it's very important that you're familiar with the light microscope because this is the one that we're going to be using in class, or we would be if we were in person. And you'll need to know or be familiar with the parts of the microscope as well as what they do. So we're just going to start and go through the numbers here. So at the top, this is the body tube. and it is connected to the eyepiece. The eyepiece, or the ocular, is right at the top. And this is where you put your eye to look through the microscope. So eyepiece, you look through this lens down the body tube, and that connects to the revolving nose piece. It's called the revolving nose piece because, one, it holds our objective lenses, but it also turns. This will spin to put these different objectives into place. So the revolving nose piece holds in place our 3 objective lenses. Now remember, we said that the magnification of our microscope ranges. The light microscope, or the compound microscope, we can change the magnification that we're looking at by changing which lens we're looking through. So we have typically 3, sometimes 4 lenses here. The lowest one is called the low power objective. You can tell the low power objective because it's the shortest one. And the magnification of that objective is going to be written on the side of it. And you can't really tell here, but if this was a color image, they're typically color-coded along the edges here. The medium size objective is called the medium power objective lens. This is going to be the one that is middle in size, so it's not going to be small, but it's going to be middle. It's called the medium power objective lens. The high power objective lens is going to be the longest or the largest of the objective lenses. That's going to have the highest magnification, and again, magnification is going to be written on the side of each of them. If we come on down, we have our stage clips. The stage clips are located right on the stage. The stage is this platform on which we sit our specimen. And the stage clips allow us to clip our specimen slide into place. So these will lift up, and I'll just place my specimen right under them, and it'll hold the slide in place. Right underneath here, we have the diaphragm. The diaphragm, it has a little adjustment knob, and it allows us to adjust how much light is coming in through the specimen. So in order to see our specimen, the specimen wants to be centered right here in the center. That way the light from the light source at the bottom can pass through. So the light here shines up. We adjust how much light goes through at the diaphragm by using the little twisty knob, and that light will come straight up through our glass slide, through our specimen for us to view. The very bottom of the microscope is called the base. When you're carrying the microscope, you want to have your hand under the base and on the arm. So the arm is here. It connects the bottom part of the microscope to the top piece of the microscope. So when holding a microscope, one hand is holding the arm and the other hand is under the base. Now when we are trying to focus our specimen, we're going to need these 2 knobs here. The coarse adjustment knob is going to be the largest knob, and we spin that to make large adjustments to how clear our image is. So we use our adjustment knobs for resolution to make our image clearer. The coarse adjustment knob is going to make large changes to how clear our image is. Once we're actually able to see our image, we use the fine adjustment knob, or the smaller knob here, to turn to make our image much more clear, but in a smaller way. So it makes smaller revisions to our resolution. So those are the pieces of the microscope. You're going to want to be familiar with those. So how you actually use a compound light microscope and figure out magnification. So the total magnification is going to be the eyepiece power times the objective lens power. Typically, the eyepiece magnification is always 10. So 10 times whatever objective you're on would tell you your total magnification. So an example, if my eyepiece is 10, my objective is 30, I multiply those together, my total magnification is 300. When you are using a light microscope or a compound microscope, when you're making your slide, you need to be sure that you cover your specimen with a cover slip, which is a thin piece of glass that you lay over your specimen to cover it. And you cover it, 1, to keep it safe, and 2, so that you don't see any bubbles, because you're going to put water on top of your specimen, and you want to put the cover slip on appropriately so you don't see bubbles. After you have your specimen in place and you put it under your stage clips, you want to start on the low power objective. So you always start looking at your specimen using the low power objective with the stage all the way down. That way you're not constantly searching, you just start with it at one extreme all the way down, and then you use the coarse adjustment knob to bring it into view. So you focus first with the coarse adjustment knob, then once you are able to see your specimen, you move on to the fine adjustment knob to make it perfectly clear. Again, total power, eyepiece times objective. Your eyepiece is most often always 10 times whatever your objective lens is. So here, this one would be 4, this one in the back is 10, and this one here is 40. So which of these would be viewed at the highest power? Which one is the highest power, and how do you know? Oh, so we didn't have an answer. This one here on the right is viewed at the highest power because I see more details. I'm seeing an individual piece and all types of fine details. So I know this one is the highest power. So our last part is knowing actually how to make a wet mount slide. Again, a wet mount slide is made in order to view our specimen. So first you got to get a clean slide. Your slides are made of glass, so you want to make sure it's perfectly clean. And you also want to grab a clean cover slip, which again is a very, very thin piece of glass. You're going to carefully place your specimen right on the slide. Remember, your specimen needs to be thin so that the light can pass through it. Then you're going to place 1 drop of water. So 1 drop of water on top of your specimen, like you see here. Then, sometimes if your specimen is clear, you might use a stain. Most of the time in our class, we're not going to use a stain, but you could to help you see things more clearly. Then you're going to add your cover slip. So you always add your cover slip at a 45-degree angle. So you touch one end of the cover slip on the specimen, you hold it at a 45-degree angle, and you either drop it or slowly lower it down on top of the specimen. You do this to prevent air bubbles. If you were to just take the two and put it right together like this, the air is going to get trapped in the water, and you're going to think you're looking at something awesome, but you're just looking at an air bubble, like right here. So again, 45-degree angle to prevent air bubbles. So here are some review questions to help you review what we learned. You should be able to answer them. So 1, what type of microscope would you use if you wanted to study the surface features of a cell? Number 2, with what objective do you start viewing with and why? And question 3, what is the proper way to create a wet mount slide? You should be able to answer those if you watch the video. Thank you.